Okay, this is the introductory material for the second part of Lab 3. So this covers the bacterial synergism portion of the lab. And this is for Thursday makeup labs that missed because of our snow day. We're going to talk a little bit about the bacterial synergism, and then you're going to discuss with your TA the experimental design for this experiment. So synergism is when you have physiological processes that are enhanced because organisms are close to each other. So I really like to think about synergism using this figure on the right. So if you think about the bars represent some kind of physiological process. So for example, it could be growth. And if you have species A, that's how much they grow on their own. If you have species B, the blue bar represents how much they grow on their own. But if you put species A and species B in the same, like really close to each other or in the same dish, for example, and you get this huge spike in growth that's much more than just the addition of those two things together, that is what synergism is. You can also think about it as like 1 + 1 = 10. So an additive effect would be 1 + 1 = 2, but a synergistic effect is 1 + 1 = 10. So that process is dramatically enhanced by those organisms, both those species being in the same petri dish together, for example. We're going to use a model organism here to investigate synergism. So just like we used a model organism, the Tetrahymena in Lab 2, we're using a different model organism here. We're using a bacteria called Serratia marcescens. This is a common soil bacterium. It's important for you to understand how synergism relates to composting, and so that's something that we will talk about with your TAs in lab this week when you look at some plates, when you look at some results for this. So I won't cover that right now. So the model system we're using here, bacteria is prodigiosin production. So prodigiosin is a red pigment, so that petri dish on the screen shows Serratia colonies that are producing that red pigment, so they're visibly red in color. And this pigment is produced by the combination of two metabolic intermediates, MBC and MAP. And so we have 3 different strains of this Serratia bacteria. One is the wild type that produces prodigiosin, so it produces both of the MBC and the MAP, the metabolic intermediates, and it can then produce this really bright red pigment. There's also 2 mutant strains, the WCF strain, which produces only the MAP, it does not produce the MBC, and then the 933, which is the opposite. It's a mutant and it only produces the MBC and not the MAP metabolic intermediate. And so in this example, for these organisms, enzymes are required to make the MAP, the MBC, and the prodigiosin. So enzymes are required to make both the metabolic intermediates and then also the pigment at the end. And so the D1, the wild type, has the instructions to make both of those intermediates, and so therefore it can make the prodigiosin. And it'll be important for you to understand where those instructions are in the cell. That's something you should talk about. You should have talked about in lecture, and you can talk about with your TA this coming week. So why can't the mutants make both of the MBC and the MAP? Why aren't they able to do that? And so this should come as no shock to you, but they don't know how. They don't have the instructions. So if they don't have the DNA, they can't make the proteins that are necessary to make those metabolic intermediates. And so the mutants don't have the instructions to make the enzymes that are needed to make one of those metabolic intermediates. This is something you also do with your TA in lab this week. It's important for you to understand how diffusion, secretion, genetics, and metabolism all relate to one another, and specifically how those concepts relate in light of prodigiosin production. So it's important to understand how prodigiosin production in Serratia works and what these 4 concepts have to do with it. And so that is discussed in detail on page 25 in your lab manual. There's a space for you to look at that, but you'll also talk about it with your TA in lab. So we're going to investigate bacterial synergism, and so your job as a group is to design an experiment that looks at that tests for bacterial synergism with these 3 strains of Serratia. So you're going to be given 3 different strains, the D1, the wild type, and then the 2 mutant types. And you'll also be given 4 tryptic soy agar, so TSA plates. So this is what you can plate the bacteria on to make them grow. So if you want to plate, for example, 2 different species on the same plate, you could do that. So your job is to design an experiment as a group and then to talk with your TA about it next week. And just so that you have exposure to this, because we're going to talk about it, and we're going to use it again, rather, in Lab 5 of this course, is aseptic technique. So you should have read about this in your lab manual, but aseptic technique is basically where everything needs to be sterile. So you need to sterilize your lab bench with ethanol. You should be wearing gloves so that you are not contaminating your samples. We have lots of bacteria on our hands, for example. But then also so that the bacterial species that you're working with are not contaminating you. So anything that you use for this lab and also for Lab 5, where we're concentrating on aseptic technique, is needs to be sterile. So any of the pipettes you use or the tryptic soy agar plates, anything else that you use like that needs to be sterilized. And so if you set something down on the bench, it's no longer sterile. So you should get a new one. It's always good lab practice to wash your hands before and after you work with any microbes, and to treat any microorganisms as if they're harmful. So even if you know they're not harmful, if you still treat them like that and you get in the habit of it, that's good lab technique. And that's it.